Hello, everyone, and welcome to today's webinar. Genome-wide CRISPR Screens in primary human T cells reveal key regulators of immune function, presented by Dr. Eric Shifrut, Postdoctoral Fellow, Department of Microbiology and Immunology, University of California, San Francisco. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. As the world leader in serving science, Thermo Fisher Scientific's mission is to enable researchers to make the world healthier, cleaner, and safer. It helps customers accelerate life sciences research, solve complex analytical challenges, and increase laboratory productivity. Thermo Fisher Scientific offers an unmatched combination of innovative technologies, purchasing convenience, and comprehensive services. To learn more about our sponsor, please visit thermofisher.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Additionally, if you are viewing this webinar on demand, you may still continue to submit questions and they will be answered via the contact information you provided at the time of registration. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Eric Shifrut. Dr. Shifrut is a postdoctoral fellow in Alexander Marson's group at the University of California, San Francisco. Dr. Shifrut completed his PhD at the Weizmann Institute of Science in Israel under the supervision of Professor Nir Friedman. In his thesis work, Dr. Shifrut developed computational and experimental platforms to map the landscape of T-cell receptor repertoires in health and disease. For a complete biography on Dr. Shifrut, please see the Biography tab at the top of your screen. Now, without further ado, please join me in welcoming Dr. Shifrut. Dr. Shifrut, you may now begin your presentation.
Thank you, Christy, for this introduction. Hi, everyone, and thank you for joining this webinar. Today, I would like to share with you what we've learned from establishing genome-wide CRISPR screens in primary human T cells. Our motivation for this project was the remarkable success of immunotherapy in recent years. I will describe some of the background information. I will go through important points to keep when performing these pooled screens, and then I will describe the results from our own screens. By way of introduction, if we think of the adaptive immune system, the problem it's trying to solve is how to specifically recognize and eliminate foreign pathogens while remaining remarkably tolerant to cells. This tolerance is achieved by built-in mechanisms, both intrinsic and extrinsic, that can allow cells to recognize foreign pathogens, but avoiding healthy tissue. Unfortunately, tumors can hijack these pathways and try to recruit suppressive cells and create an immunosuppressive niche, and by that, escaping deletion by the immune system. Immunotherapy has taught us that there are ways that we can manipulate cells and manipulate their environment and how they sense these signals to ignore suppressive signals and create an effective response against tumors. While very promising, there are still many challenges that remain in current available modalities of immunotherapies, and we are thinking of ways to engineer cells to resist these immunosuppressive niches. Our overall goal is to try to find ways to reprogram cells to ignore or translate these suppressive signals actually to an activated state, creating effective, long-lasting cures using engineered cells. The way we go about learning the ways of how to engineer cells is through these unbiased large-scale screens. So overall, two types of genetic screens are in existence, pooled library screens as well as arrayed library screens. These are very powerful tools for mechanistic discoveries and performing data-driven research. In general, arrayed screens are perhaps more simpler to execute. In a screen like that, a plate would have a population of cells in each well. Each well will have its own perturbation, a gene knockout. This could be either with SLICE-SERNAs or CRISPR-based. But now this plate can be replicated and used for downstream analysis. Arrayed screens, as I mentioned, perhaps are more simple to execute, but they do not scale well. If you want to explore more gene knockouts, you will need to perform and have more plates in your assay. On the other hand, pooled screens are very easy to scale, where all cells essentially live in the same vessel. Each cell will have its own perturbation depicted by a guide that is integrated in that cell by a lentiviral particle. After selection is applied and cells are pelleted, genomic DNA will allow us to perform next-generation sequencing for the guide distributions and ask which guides are enriched before and after selection. An essential component of pooled CRISPR screens is the ability to have this integrated barcode. Overall, when trying to establish a pooled screen, one needs to think about the various steps. So the first step is to introduce a library of guides. This library of guides will be used to generate a pool of lentiviral particles. The library itself can now be obtained either from a public resource such as Agene, or if you're interested in a targeted gene pool by using custom synthesis and cloning the plasmid library in-house. To design these guides, many of the rules have been already figured out. One example for a very powerful tool is the CRISPR tool, which is available online. Once the lentiviral particle pools are generated, they can be concentrated, titrated, and then frozen in aliquots, and this will allow for consistency between different replicates of the screen. One thing to maintain, and it's very important to note in pooled screens, is that the representation of the library or coverage, meaning how many cells for each guide in the library exist during every step of the screen, is maintained at a high enough number. Overall, that number can range between 500 to 1,000x, and this requires a large number of cells. So when you go about doing screens in your own cell type, the important thing to take is what is the scale that you're comfortable culturing your cells in. Because this coverage is important, here I'm showing from a recent review by John Dunch example numbers for a screen. So if the size of your library is about 100,000 different guides or different perturbations, and you want each of them to be represented at least 500 times in your library, this means that you need 50 million cells that are positively infected and expressing the guide. Transduction efficiency using these lentiviral particles is never 100%. So assuming about 40% inspection efficiency, this means that you need to start with 125 million cells. On the right, we can see a plot that shows that if coverage drops below a certain threshold, the noise that is produced at the end of the screen is so large and basically damages the signal-to-noise ratio and inhibits our ability to robustly detect our hits. So although pooled screens are very powerful, so far they've been limited only to cell lines that express the Cas9 stably, or in transgenic mice also that express Cas9. As a quick reminder, the CRISPR system requires two components. One is the Cas9 enzyme, and the other is the guide that will direct the Cas9 into the location in the genome. A double-stranded break will happen, and this most likely will be repaired by an error-prone mechanism, resulting in a gene knockout. So for Cas9 RNPs, we can use a synthetic Cas9 protein and a synthetic guide. These will be assembled ex vivo and will be introduced to cells with various methods. It could be liposuction. In our case, we tend to use electroporation. However, this does not allow us to do a pool screen because the Cas9 and the guide will be eventually degraded, and we will have no record of what perturbation that cells undergone. On the other hand, as I mentioned, to try to introduce the Cas9 into the entirety using lentivirus is quite ineffective due to its very large size. So to establish pooled screens in primary human T cells, we decided to take a hybrid approach. In this experimental timeline depicted at the top, we will introduce the guide with the lentivirus. This lentivirus is compact and will be able to transduce cells quite efficiently. The Cas9, which is harder to introduce by a lentivirus, we will introduce by electroporation. Our hope is that Cas9 will meet the in vivo transcribed guide, will assemble, and will result in an effective gene knockout. We call this approach SLICE for guide RNA lentiviral infection with Cas9 electroporation. To test the feasibility of this approach, we used an example guide, the CD8 alpha receptor that is highly expressed on CD8 cells. And as a control, we also expressed a guide that is called a non-targeting control, a sequence that does not appear in any known region of the human genome. On the green distribution across two donors, you can see that expressing the CD8 targeting guide effectively knocks out the CD8 alpha protein in a majority of cells, whereas the cells expressing the non-targeting control at the gray distribution maintain wild-type levels. To test if this is generalizable, we also tested a different target, CD45, as I mentioned, across donors, but in this case also across different cell types, CD8 T cells and CD4 T cells. And in both cases, we can see that the majority of cells expressing the CD45 targeting guide lose expression of the protein, whereas the non-targeting control, as I mentioned, retain their high level of expression. This proof of concept showed us that we could perform SLICE, or basically using a one guide library, create edited T cells with a traceable perturbation that can be later read out with next generation sequencing pipeline. So now, going from one guide through a series of optimizations, we scaled up our library to a genome-wide CRISPR knockout library. This library was obtained from Angene and is termed the Brunello library. In our case, our biological question was trying to find regulators of human T cell proliferation in response to TCR stimulus. So following the SLICE protocol, at day 14 of culture of cells, we reintroduced them to TCR stimulus. Before this stimulation, we also stained them with CFC, or a cell proliferation dye, that will allow us to track the division of cells and perform a sort that will enrich for dividing cells and non-dividing cells. Later on, using next-generation sequencing, we will count the abundance of guides in each of these sort bins, and we'll obtain for each guide in the library a log to fold change, showing us whether these guides were enriched in dividing cells. These will have positive values, or depleted in dividing cells. These will have negative log fold change values. So in this representation, I'm showing the distribution of log fold change in the entire library for 3 example positive hits and 3 example negative hits. In this representation, each line is a guide targeting the gene that is depicted on the left. And for these hallmark negative regulators of TCR signaling, CBL-B, CD5, or U-3A, you can see that multiple guides targeting the same gene are showing enrichment in the dividing cells. Whereas on the other hand, essential components of TCR signaling, VAV1, CD3 delta, and LCP2, are all showing a depletion from the dividing cells and enrichment in the non-dividing cells. This is another advantage of a pool screen because we have redundancy not only of cells per guide, but also of guide per gene. And this increases our confidence in the hit where multiple guides targeting the same gene are showing the same trend. We can now look at all hits in our library, and in this volcano plot, on the y-axis, you can see how significant the hit is on the gene level, and on the x-axis, you can see if it's either a positive or a negative hit. So on the left side, we can see that many essential components of TCR signaling, a few of them we already mentioned, LCP2, CD3 delta, and VAV1, are shown to be enriched in the non-dividing cells. The more maybe therapeutically interesting part is the right hand of this plot, showing CBL-B, U-3A, many others that were described in the literature, but also a large number of novel regulators. To show that our screen is reproducible and these hits are not a result of some chance or off-target effects, we performed the same screen again with 2 new donors at a genome-wide level and asked whether these hits are now appearing in both screens. So in this representation, I'm showing the normalized log4 chain score of the first screen on the x-axis and for the second screen on the y-axis. So things that fall on the diagonal are actually reproducible between these 2 screens. And many of our hits, as I mentioned, seem to be hits in both screens, increasing our confidence that these are true hits. To try to annotate this collection of genes, we took the gene set enrichment analysis approach, trying to find pathways that are enriched in both sides of the screen. On the bottom right, we can see that many of the essential TCR signaling components, as depicted by the KEGG pathway database, are indeed depleted or receiving negative log4 chain scores in our screen. On the right red box, we can see the top candidates for that. Again, LCP2, VAV1, but also CD3 delta, LCK, and others. On the other side of the screen, genes that, when they're knocked out, actually increase the ability of cells to respond to the TCR signaling by increased proliferation. There was no literature, there was no bioinformatic database that collected all of them under the same list. But through literature search, we found an SHRNA in vivo screen that was done in mice in a tumor model where OD1 T cells are introduced with a library of shRNA, and then they're allowed to infiltrate the tumors. The authors asked which guides allowed cells to survive in the tumor tissue. To our surprise, and we're very content, many of our negative regulators that were found in the genome-wide CRISPR screen in human T cells were also hits in the in vivo mouse screen. So CBL-B, SOX1, BGKA, and Z, June were all found both in our screen, but also in the in vivo screen done in mice, targeted towards finding immunotherapy hits. Now that we have a list of candidates and hits from our screen, we aim to validate them and see that they reproduce their function also in an orthogonal system. For this, we took the second approach for genetic screens, which is array screens. In an experimental timeline like that, we take CD8 T cells from different donors. We introduce gene knockouts by using synthetic Cas9 assembled with synthetic guide RNAs in a plate. These are assembled and introduced to T cells by electroporation. Now we create arrays or plates of edited T cells, each well having a homogeneous population of cells with a single gene knocked out. For each gene in our list of candidates, we use 2 different guides. Now these plates can be used for downstream phenotyping analysis, both to validate their ability to increase proliferation based on CFSC, but also to look at early activation events and see if they're affected by the gene knockout. Here I'm showing our top 2 candidates, CBL-B and CD5, in the top 2 rows, across 2 donors and 2 guides directed at knocking out these genes. And the CFSC distribution in the green shows us that CBL-B and CD5 increase proliferation in response to the TCR stimulus, as the non-targeting control in the 3rd row from the top shows very little proliferation. On the other hand, LCP2, which is the central gene for TCR signaling, when this member is knocked out, cells are completely unable to respond to the TCR stimulus, and they remain non-dividing, same as the unstimulated cells in gray across all panels. To quantify the ability of hits to increase proliferation, we fitted the CFSC distribution using Gaussian curves. This allows us to extract a quantitative parameter called the proliferation index, which is an indicator for how strong cells proliferated. When we normalize this proliferation index to the non-targeting control, we can see that many hits from our pool screen as well validate their ability to increase proliferation in the array screen. We do note that some of them do not validate, and these are interesting by themselves, and we're following up on them. As I mentioned, array screens allow for multiplex phenotyping, and not only that we can look at CFSC a few days later, we can also look at what are the possible mechanisms that are driving this increased proliferation. So one hypothesis is these events are driven by increased activation state. We use 2 exemplar protein noted for their upregulation during early events downstream of TCR stimulus, CD69 and CD154 or CD40 ligand, and looked at their ability to increase activation state 6 hours post-exposure to TCR stimulus. Again, we are normalizing to the non-targeting control, the second from the bottom. And overall, many of our hits from the screen are actually inducing heightened or elevated activation state for the same time point compared to the non-targeting control. Again, our essential component LCP2, when it's knocked out, disables the cells from even going to the activated state. Another form of what we can do with these now plates or arrays of edited T cells is basically to look at their ability to interact with other cells. In our case, the relevant context was their ability to kill tumor cells. To do that and to introduce an antigen-specific kill, we also needed to introduce the transgenic TCR. In this case, we used the model system of the 1G4 TCR that recognizes the NY-ESO peptide in an A2 context. So we will, in this experimental timeline, we will introduce the TCR with a lentivirus. We will then edit the cells with Cas9 RNPs. We will co-culture them with 8375 melanoma cell line that also expresses an RFP in its nucleus. This RFP will allow us to count cells using live cell imaging and look at either the proliferation of the melanoma cells or their killing by the T cells. To see that the system works, we first show that we're able to overexpress the NY-ESO specific TCR in primary immune T cells across 2 donors and at very high efficiency. As a control, we also looked at if the TCR or the T cells are killing A275 melanoma cell lines in this co-culture model in a specific way. So we see that there's also a dependency on the ratio between the A375 cell line and the CD8 cytotoxic T cells. As we introduce fewer and fewer T cells, the A375 are allowed to grow, and this is depicted on the bottom 2 panels. The upper panels shows us that this killing of A275 by T cells is dependent on caspase activity, showing that this is a true cytotoxic functional assay. Looking at the cells, the responding cells, the cancer cell line, 36 hours after co-culture, if we don't introduce any T cells, this is in the A375 only panel, cells grow uncontrollably. When we introduce T cells that are specific to A375, but are knocked out for non-targeting control, there is some clearance of the tumor cells. However, remarkably, when we introduce 4 of our candidate hits, RASA2, CBL-B, FOX1, and TCB2, we can see that the clearance is much higher, indicating that these engineered T cells now have a boosted ability to clear the tumor cells. To quantify this over time, we can look at the count of A275 cells throughout the culture, because this plate is now under continuous live cell imaging monitoring. So whereas the non-targeting control cells, as I mentioned, are able to clear some of the tumor cells, these 4 candidates, TCB2, SOX1, and CBL-B, and RASA2, are able to clear the tumor cells much faster. As the counter example, LCP2, its knockout disables the T cells from recognizing even the A275 and allow the A275 to grow uncontrollably. So we were very happy with this pipeline or discovery platform that we've established here. We've done unbiased genome-wide screens in a pooled fashion in primary immune T cells. We took the candidate hit, validated their ability using array screens, but also looked at their functional capacity to kill tumor cells using the live cell imaging co-culture assay. As additional development, we can supplement the discovery power of pooled screens by not only using TCR stimulus, but also using a suppressive force. If you remember, our original motivation is to try to find ways to engineer T cells to resist the immunosuppressive niche that is created by tumors. One way to mimic this that we went about is using an adenosine agonist. Adenosine is in very high levels in the tumor microenvironment and acts as a suppressor of T cell proliferation. So in our case, we wanted to ask the question, can we find through these genetic screens perturbations that will make cells resistant to adenosine signaling? So in the same way that I've described before, we've performed this genome-wide screen, but now we also added an arm where we add a mimic of the adenosine to a signaling. And this is a synthetic inhibitor, and it can be added easily to the media. Using the same approach, we looked at whether this synthetic inhibitor, CGS-21680, does indeed inhibit the cells from proliferating. And you can see based on the CPC distribution, there are definitely fewer dividing cells compared to the vehicle control arm. Now, same way as before, we can take these two sort bins of dividing and non-dividing cells and ask, can we find guides or gene knockouts that allow cells to resist the presence of the agonist? In this plot, I'm showing you the log fold change normalized on the x-axis where the agonist is not introduced. And this is just a vehicle control. And on the y-axis, we're showing the log fold change when this adenosine agonist is added. I did mention that this agonist is specific to the adenosine A2A receptor. And we were very happy to see that right on the y-axis, you can see that the gene ADORA2A that encodes for the A2A adenosine receptor is specifically enriched in the only when we add the adenosine agonist. So this is a nice positive control, but also we could find a previously uncharacterized protein 515A, and this is the first time it's been associated with response to adenosine signaling. We took the same approach and validated both these hits, the A2A ADORA receptor and also FAM15A, showing here across two donors that they're actually specific only to the presence of the adenosine agonist. We took this even further to our co-culture system, and we can show that while the non-targeting control cells are inhibited from killing or clearing the A375 melanoma cells, the knockouts for ADORA2A and also our novel hit FAM15A are able to resist the effect of the high adenosine agonist and maintain wild-type killing levels. So this is one way of enriching the type of information that you can take from these pooled screens. And the last platform that I'll share with you today also allows for enriching the type of data that you can get from a pooled screen. So in recent years, both PROTRUB-seq and CROP-seq have emerged as ways of coupling a pooled CRISPR screen together with single-cell RNA-seq output at the other end. This now allows for these very high rich data sets that allows you to compare a pool of guides and their downstream effects at the transcript level. To achieve this, one needs to capture the guide sequence using any single-cell RNA-seq platform. However, the guide RNA, is, because it's transcribed by a Pol3 promoter, does not have a poly-A tail. The CROP-seq platform that was published about 2 years ago solved this in a very clever way, where it's integrating the guide cassette within the LTR region that eventually will be duplicated once it's integrated into the genome. This creates two cassettes of the guide RNA, one that will be used for the genome editing, but the other one will be transcribed by the AF1 alpha promoter, having eventually a poly-A tail. This will allow it to be captured by any poly-BT downstream platform. In our case, we use the 10x genomics platform. As I mentioned, this now creates a very rich data set for every single cell. Not only will you get its transcriptome, but also which guide was expressed there. And you can start ask questions of what is the relationship between a perturbation and the downstream effect. So we performed our SLICE platform, but now instead of sorting cells at the end, we actually submitted them using because we used the proper CROP-seq vector in a targeted library with 48 candidate guides. We can use downstream single-cell RNA-seq and try to now explore in what ways our perturbations are increasing cell ability to activate and proliferate in response to stimulus. I should mention that as a control arm for this experiment, we also took cells that we introduced the library to, but did not expose to a TCR stimulus following. With single-cell sequencing, you tend to get these like high-dimensional maps that describe the gene expression across all cells in your library. So in this depiction here, we can see that there are regions in this high-dimensional maps that are more associated with quiescent cells or resting cells. This is depicted by high expression of the IL-7 receptor, for instance, and the CCR7 receptor. No, good. Chemokine, sorry. On the other hand, on the right hand side of each of these high-dimensional maps, we can see the highly proliferating cells indicated by Ki67, which is a cell cycle gene, but also their cytotoxic ability is increased, depicted by granzyme B. Now that we have this gene expression map, we can start asking, are they related to the perturbation the cells have undergone? As a reminder, each cell now, not only that we know that it's gene expression, but also which guide was expressed there. So for our two top candidates, it will be in CD5, we can see that they're clustered. All cells that have CBL-B and CD5 are clustered in the right-hand side of the this high-dimensional map, exactly where proliferating cells fall. Where LCP2, the negative hit, pushes cells towards the left hand side of the plot, where the resting cells are. The non-targeting control are evenly distributed across this high-dimensional map. We can now create a map of cell states by subclustering cells based on their gene expression and ask whether guides fall together within the same cluster. And overall, we can now start digging deeper into this type of data and try to learn which guides or which perturbations tend to push cells to have a more common cell state. Now, so for instance, in this example, clusters 1 and 2 are associated with resting cells, so we can see depletion of CBL-B and CD5 and CDK and 1B and so on from these clusters, and we can see enrichment of the LCP2, CD3 delta guides. Clusters 10, 11, and 12 that are associated with proliferating cells have various combinations of the perturbations enriched in them. So to summarize what I've shown you here today, we've established a very powerful discovery platform that allows us to do unbiased genetic screens using the CRISPR-Cas9 system. And as an example, we showed this in primary human T cells, and more specifically in CD8 T cells, asking the question of, can we describe the regulators T cell proliferation and response to TCR stimulus? We use this assay to screen, as I mentioned, and find guides that will increase the cell's ability to function and proliferate. We validated the hits that we've got from this screen and also showed that their knockout can increase the cell's ability to kill cancer cells in this in vitro culture system. And with the adenosine agonist example, I've shown you that a suppressive force can be added to the pool screen to try to explore now mechanisms that make cells resistant to inhibitory signaling. So I will end just by showing you an overview of the SLICE platform. As I mentioned, now this enables these large-scale unbiased genetic screens in primary immune T cells for the first time. You can use any commercially available off-the-shelf guide RNA library. And Cas9 is introduced by electroporation. This now allows you to explore many different biological functions of T cells. We chose to focus on T cell proliferation using the array screen part of this platform. You can now start asking ourselves actually proliferating and activating more strongly when these genes are knocked out, and you can also perform these co-culture assays. Finally, I showed you that using single-cell RNA sequencing, you can now create these like high-dimensional maps and create relationships between guide perturbation and downstream cell state. I will just end by acknowledging the people involved in this project. First, Julia Carnevale, who's a clinical fellow in the Ashworth Lab here in UCSF, and my mentor, Alex Marson, and members of the Marson Lab that helped with performing these screens. I will thank our funding resources. I want to thank everyone for their attention, and I'm happy to take any questions. Thank you.
Thank you, Dr. Shifrut, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box and click Send. We'll answer as many of your questions as we have time for. Okay, our first question. Dr. Shifrut, can the SLICE protocol you described be adopted to perform genome-wide screen with other types of cells, such as neuron cells? And what are the potential challenges?
That's an excellent question. So in essence, the two requirements for performing genome-wide screens in this SLICE platform is one, that you should be able to transduce your cells with lentiviral particles. And the other, you should be able to grow them in large numbers. Actually, the number of cells that you're able to culture comfortably determines the size of the library. So in a case where, I don't have experience with neuron cells specifically, but in a case where the cell number is a limiting factor, using the coverage math, we calculated back to what type of, or what is the size of the library that you can accommodate in your screen. So once an electroporation protocol is found for your target cells, and you know that you can transduce the cells, essentially you can perform a slice to any type of primary cell. We've shown it to work also in other type of T cells, as I showed in the beginning, but we're now also exploring other immune cell types. I would say that lentivirus is thought to be able to transduce also quiescent cells. So it could be that neuron cells can express a guide out of the lentiviral cassette. Yeah.
And Dr. Shifrut, how many SCRNAs are used per gene for genome-wide screen?
So I would say that generally the number you try to keep is around 500 to 1,000 cells per guide in your library. And the libraries that we've used, as I mentioned, Brunello library that is designed by John Dunch and is available on AdGene, they chose to use 4 guides per gene in the library. This number ranges between, I've seen libraries with 4 guides, the most compact, up to 10 guides per gene. As I mentioned, the size of the library also determines the number of cells you'll need to culture. So I would be careful not to increase the size of the library just to have redundancy of guides. In our experience, the 4 guides per gene, if you maintain sufficient coverage, is enough for many biological systems.
Now, could you please share how to minimize off-target impact based on your experience? Yeah, so when the CRISPR-Cas9 systems were starting to be more and more popular, it was obvious that some of the ailments of the earlier perturbation platforms, such as SHRNAs, were less of a concern in the CRISPR system. It's fairly robust, and off-targets are quite minimal. The nice thing about the full screen, as I mentioned, because of the redundancy that occurs at several different levels, both many cells express the same guide, and there are multiple guides targeting a gene. And in our case, we also performed the screen in multiple donors and multiple times. The effect of
off-target cutting is reduced to basically negligible levels. So I wouldn't worry too much about off-target effects in these commercially available screens, libraries, sorry. And I would also know that for every screen there will be a validation phase that follows, and that this allows you to use different guides for the same gene, not necessarily the guides that were present in the library, really reducing the off-target effect.
Thank you, Dr. Shifrut. And it looks like we're almost out of time, so this will have to be our last question. What software algorithm was used for SLICE with single-cell RNA-seq data analysis? And could you please specify how to perform data analysis?
Yeah, I'm happy to. It's a rather complex system, I agree. So first, you need to be comfortable with analyzing single-cell RNA sequencing data sets to begin with. The tools available for that are now in very active development, and people gain more and more experience. The field is getting better at creating platforms. We used a combination of a publicly available powerful package to analyze single-cell RNA sequencing called SURAT. And this, I recommend this package for every type of single-cell RNA-seq data set. The complexity that is added with CROP-seq or PerTurb-seq is that now for every cell you also have an identity tag that tells you which guide was expressed there. In our approach, we took a very straightforward algorithm. We just used the chi-square test to ask whether we can find an enriched association of guides in certain clusters. I think as these, again, PerTurb-seq or pooled screens coupled with single-cell RNA sequencing datasets become more and more popular, there will be dedicated tools and algorithms that will be tailored to the type of assay.
Thank you, Dr. Shifrut. Now, do you have any final comments for our audience?
Yeah, thank you all for listening and for these insightful questions. I'm available by email if anyone wants to contact me with follow-up questions, and I wish you good luck.
Thank you again, Dr. Shifrut, for your time today and your important research. And we'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, a final thank you to our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks for joining us, and we hope to see you next time. Goodbye.